All right. Thank you for the opportunity to present today. The focus of my presentation today will be about insulin signaling modeling using multi-omics data. This is the focus of my team. We kind of do a lot of informatics like everyone else here. But today what I will do is I'll just focus on the neuroinformatics and a couple of methods we developed in that area. Here's the outline of my talk. Okay, I'm just trying to see there's something else coming up on my screen here. Here's the outline of my talk. First, I'll go through the two manuscripts I presented, we published recently. And then I will also talk about the insulin signaling pathway and how it impacts the expression of one of the gene, especially an inflammation marker in Alzheimer's disease. And then I'll talk about the simulations that we have done after we build the model with some in vitro data as well as some patient-specific data that we found in the public repositories. And then I'll further give some conclusions. So as you all know, the biggest difference between omics data analysis and the mechanistic models are one is data-driven, which I'm quite familiarized with because majority of my research focuses more in the pharmacogenomics, precision medicine, and cancer informatics. So where we are looking into statistical models and then trying to identify biomarkers. Versus, as you know, the mechanistic models, it's more based on a particular mechanism, and then we build these ODEs. So a little bit background about the blood-brain barrier. The critical function of the blood-brain barrier is it involves cerebral blood flow, energy metabolism, and immunomodulation. And these functions are regulated by these insulin signaling pathways primarily. The endothelial insulin resistance. Okay, I'm not able to see properly here. Okay, that's fine. Endothelial insulin resistance can lead to the BBB dysfunction. And that's primarily in the diabetes patients, insulin resistance also leads to ADs or hypothesis. And then the molecular mechanisms that are regulating to the neurovascular unit composition are poorly understood right now. So our main aim is to understand the dynamics of insulin responsive pathways, specifically focusing on the endothelial cell lines and also using the patient data here. So a little bit background about these two manuscripts I just said I will be describing very briefly. These are published manuscripts. We are the first group to publish the blood-brain barrier transcriptomics data in such a deep depth. So I think we published this is one cell line, human endothelial cell line, with more than 900 million reads. So you can see how depth we went to kind of understand the one single snapshot of this particular cell line and its transcriptomics data. We did submit this data out into public. It's already there on the website. And we can identify the coding, non-coding regions, non-coding genes, if they're expressed or not, as well as circular RNAs and long non-coding RNAs. And also we could also see if the pathways, if you're interested in a particular pathway, if that particular pathway is expressed or if the genes in the pathway are altered or expressed. We kind of also did a microRNA experiment on the same cell line and also published in the same blood-brain barrier omics database. Later on, I'm just trying to show you why we built this, why we started doing the systems modeling. First, we built the database, and then we learned a lot about the human endothelial cell lines, blood-brain barrier specifically. And then we went on and as this is kind of already known that the insulin signaling pathway is associated with cerebrovascular pathologies in AD, including this inflammation, BBB disruption, and A beta deposition. Using multiple genes here, for example, AKT, and then GSK3 beta or a few other genes listed here. So they could be responsible for these functions. So we and several others have published this. But as I said, there was only a single snapshot of BBB omics data we had. We still wanted to understand the insulin dynamics that's happening at the blood-brain barrier level. So one first experiment, what we did is we did a flow cytometry experiment. And then we were trying to look at the untreated and treated insulin data, and then compare the live and dead cells here. And we did not find any big differences. The ratio did not change, and it was pretty similar. Then we did a time-dependent experiment where we were looking at the Western blot data, and then we tried to see if the IR beta expression changes. We saw that it kind of goes down as like at 300 minutes, as like time increases, the insulin receptor kind of goes down. We did that in the Western blot. We also saw that in the gene expression as well. So then, since there is no dynamics data, we went ahead and then did the RNA sequencing of these 5 time points. So as you can see here, one is like a plate that's like a control, and then this is an insulin-treated HMEC-D3 same cell line. So we did that at 10 minute, 20 minute, 40 and 80 and 300 minutes. And then we kind of extracted the RNA and then did the sequencing of these samples, did the quality control, and then we fed the data into the short time series expression miner, this particular method. And then what this method does is it identifies the clusters of genes that are following the similar pattern, but with distinctive and representative expression patterns, basically. There are a few parameters you have to play with, and then it kind of gives you those sets of genes that are following similar patterns. So what happened is when we did this, we identified 12 significant clusters with about like some 6,000 genes. Of those, we picked primarily 3 groups. One is like right after insulin treatment, this particular group, the set of genes in this clusters are going up. And in this, at early time point, they go up, but then later on, they're going down after insulin treatment. And then in the third group, we found that the genes are downregulated in general. So these are the top pathways associated with these particular groups. And as you can see in the next slide, I kind of summarize like specifically the group 1 is upregulating the matrix metabloproteases pathway. And then the second group, the one that had a high peak at the early time point and then went down at the later time point, had the insulin receptor signaling pathway that was upregulated. And the third one that had like, the set of genes that were downregulated were associated to the interferon signaling pathway. Kind of more makes sense here. So then, it's like as a first start, we started looking into the insulin receptor signaling pathway. It totally makes sense that it gets upregulated and then kind of goes down. It kind of agrees with our previous Western blot data too. So we started looking into this because insulin resistance and blood-brain barrier connection, we know that, right? So we started looking into that. And then our goal here now is to build a model for this insulin signaling pathway. But then what is the outcome we want to see? Because this is a cell line, and what is that? It's not like we are more used to like a cancer cell lines and like cancer data. But here, when it comes to AD, Alzheimer's disease, it's different, right? You're looking at cell death and a few other things. And, we were just wondering what would we look at? But then we found out like we could be looking at this particular gene, BCAM1. It's an inflammatory marker that's expressed on the blood-brain barrier endothelial cells, on the cells. So then we thought like, since it's also an early diagnostic marker for AD, we'll focus on this one as an outcome. Then we started like simulating this. I'll just show you in a bit here. And then looking at different like insulin resistance, A-beta exposure and all that in this particular cell line. So this kind of strategy we follow like everyone else is like, first construct this model. And then, after we constructed the ODEs, then we wanted to identify the parameters using the MATLAB and SynBiology. This is a program we used in our case. I'm sure there are other programs, but this is what we did. And then we gave our proteomics data as the initial values for these ODEs, and then tried to simulate the BCAM1 expression. So here is the data. So far I talked about the gene expression, and then now we wanted to see if we can take this into the next level, to the protein level. So we went off into the lab and collaborated with Dr. Kandimala and Zhentao Wang, who has done a lot of the studies that I'm presenting today. So basically, what Zhentao did was like he looked at in kind of like dose-dependent insulin manner here, like from 1 micromolar to 1,000 micromolars. He was looking at like what happens to the phospho-AKT and ERK phospho-ERK data in the Western blots. And then what we found was like, you can see like, you know, the phospho-AKT kind of increases slowly with dose-dependent fashion, whereas the ERK only kind of like gets activated at the superphysiological concentrations. That's about 100 nanomolar. So with that, we kind of understood at normal concentrations of insulin, it activates the AKT, and at the superphysiological levels, it activates like both AKT and ERK. That was what we came up with after this Western blot and experimental data. And then obviously we wanted to connect our story to the inflammation marker, the VCAM, right? So we also looked into like what happens when at 1 nanomolar and what happens at the 50 nanomolar of the insulin. We found that at 51 nanomolar, the VCAM1 is not expressed. At 50 nanomolar, the VCAM also is expressed. So that kind of like gave us more confidence into like digging deeper into this particular pathway, the insulin receptor signaling pathway. And this particular pathway is composed of 2 arms, AKT, as well as like displayed in blue color here, and the ERK pathway that's displayed in red color here. And then for us, we thought this is outcome, right? The VCAM1, and it's a function of the AKT phosphorylation as well as the ERK here. So the degradation and then the synthesis kind of thing here, right? And then the degradation here is like in a linear fashion. That's what we are seeing. We saw in our previous Western blot data that it follows a linear function with the PAKT concentration. And then the synthesis one kind of follows the Emax, the dose-dependent function is what we found. So then VCAM is a combination of these two, so the degradation and synthesis. So with that model, we went and then tried to fit the data using the 1 micromolar concentration data. And our model predictions agreed a lot with the experimental data, as you can see here in the Western blot, as well as in here as well. So at the beginning, the AKT took off, whereas the IR and VCAM1 expression, as expected, were kind of going down with our model. And then with the 50 nanomolar data, we saw the same opposite what we were seeing previously in our model. So basically, the AKT went up, and then the ERK also went up, and then the VCAM also went up. So this kind of agreed with what we had in our previous model when we built the model. So as I said, we are very much used to the cancer genomics, biomarker and those kind of analysis that we do normally. So we tried to follow a similar path here. There's a lot of manuscripts out there where, we build the models first, biological models, and then fit the kinetic parameters, which we have done now. And then, in cancer, like they take the cell line data from different cancer cell lines and then use those as initial conditions. Since we don't have that many cancer cell lines, we took our endothelial cell line, the same cell line, and then treated it with different A-beta and A-beta 40. These are the primary isoforms that are observed. So we used those, and then we were looking at we generated the phospho array, the proteomics array data, and then looked at the AKT and ERK pathway, signaling pathway proteins. Here, this is the protein data. And then we, as expected, we saw that the AKT pathway is down and then the ERK pathway is up in both A-beta 40 and 42. This is the summary of these particular proteins from the RPPA array assay when we looked at the A-beta versus the control. And then looking down here, this is the time-dependent data. And as you can see, like at 1 nanomolar insulin, the VCAM expression is very low, like expected. And when it's treated with A-beta 40, our VCAM1 expression went up a little bit. And when at the higher, insulin values for 50 nanomolar, the superphysiological conditions here, it is like the VCAM expression also increased. And with the higher insulin as well as the A-beta 40, again, the VCAM went up even more. So we looked into the same here, like in the model prediction, we did the same for the A-beta 42 data, and we observed similar things like with the higher insulin, dose, we always saw that the VCAM expression was high. Then we also took some publicly available data. As I said, this is an endothelial cell line, which normally if you take any patient's data, like from, publicly available data, like websites or data repositories, you will see bulk RNA sequencing data, which, will not fit what we are looking at here, because we are specifically focusing on the blood-brain barrier, which is enriched with the endothelial cells and pericytes. So what we did is we found another data set from Vice Corey's lab from Stanford, where they published the single cell data. And then we saw that they had like endothelials and pericytes enriched here. So we took the endothelial data and then tried to fit that single cell data into our model. And again, he had the data for like Alzheimer's disease as well as the control. And like expected, we saw for the A-beta positive, which is the patient's data, we saw the VCAM was high. And for the A-beta negative, which is the control data, we saw that the VCAM expression was low. But as you see here, there are a few outliers. But then when we go back and see 19 expression data, it kind of agrees that these 83 and 84 and 86, they have the low VCAM expression. Indeed, it kind of agrees with our predictions. So in conclusion, we have done the time series transcriptomic analysis and identified these pathways. And then we build as I said, like in the lower concentrations of insulin, the AKT gets activated and then VCAM is low, versus in the higher concentrations, the ERK gets activated as well, and then the VCAM expression increases. We kind of saw that phenomena and then built the model and then tested that using the proteomics and transcriptomics data that's available outside, using both in vitro and as well as the AD patient data. So obviously, without the NIA and NIH funding, this work is not possible. And then I would like to thank the members of my lab as well as Dr. Kandimala's lab, specifically Zhentao Wang, who has done a lot of experimental work, as well as in my lab, we have done like more informatics work, Dr. Sharjah Tang and Dr. Kevin Townsend. So thank you all. We'll take any questions.
Do you have any questions for our speaker? No, maybe I have just one. You have shown that in the two different top conditions, there was one component of the MET pathway that was always negative, while the majority was positive, increased. There was one that was always decreased. Do you know whether that has some meaning or? Yeah, in the next slide, I think is the next slide. So the ERK pathway, you have everything that is upregulated, but then you have always that is always downregulated. Has this component a meaning in this pathway, or is it?
No, not really. So again, this is like RPPA, the proteomics array we did here, and we were looking at the data, and then this is what we found, and we were trying to fit with the rest of the model that's out there, primarily looking more into the ERK. As you can see in this equation, we have the phosphorylation ERK and then phosphorylation AKT that we are more interested in.
Okay.
So it did not really bother to us that the particular one was different. Yeah.
Thank you very much.
Yeah.
Okay, let's thank again our speaker.